Cognitive decline is not just about memory loss. It represents a major public health challenge. Dementia and related cognitive disorders are now among the leading causes of death in the United States. According to U.S. mortality data, dementia was the underlying cause of more than 288,000 deaths among adults aged 65 and older in 2022. However, these numbers likely underestimate the true impact of the disease. Alzheimer's disease and other dementias often contribute indirectly to death through complications such as infections, frailty, or other medical conditions. When researchers account for these broader effects, some analyses suggest that dementia may contribute to more than 13% of deaths among older adults in certain populations. For this reason, understanding the molecular mechanisms that drive cognitive decline is not only a scientific challenge, but it's also an urgent medical priority. One of the major reasons cognitive disorders are becoming more common is the demographic change. If we look at the population pyramid from 1880, we see a triangular shape. This type of age structure is typical for a rapidly growing population with many young people and relatively few older adults. By the mid-20th century, the structure began to change. The baby boom that followed World War II created a large cohort of individuals born between 1946 and 1964. At the same time, improvements in medicine, nutrition, and public health allowed more people to survive into older age. As this generation ages, we actually watch the demographic bulge move upward through the population pyramid. By 2020, the United States has a much larger proportion of adults over age 65 than at any previous point in its history. This demographic shift has major implications for diseases associated with aging, including Alzheimer's disease and other dementias. Today, roughly 7 million Americans are living with Alzheimer's disease or related dementias, and this number is projected to nearly double by 2060 as the population continues to age. Another striking statistic is that about 1 in 3 older adults will die with Alzheimer's disease or another form of dementia. Because dementia risk increases dramatically with age, the growing elderly population means that cognitive disorders are becoming an increasingly important medical and social challenge. This slide shows the leading causes of death in the United States in 2023, separated by men and women. As expected, heart disease and cancer remain the 2 leading causes of death overall. These diseases have historically dominated mortality statistics in developed countries. However, if we look further down the list, we see that Alzheimer's and other dementias now rank among the top causes of death in older adults. Dementia is somewhat unique compared with many other causes of death because it's not always recorded as the direct cause for mortality. Instead, patients often die from complications such as infections, malnutrition, or injuries that occur as cognitive function declines. Even with this underreporting, dementia, driven primarily by Alzheimer's disease, but also including vascular and Lewy body dementias, consistently ranks within the top 10 causes of death in the United States. This highlights how significant neurodegenerative disease has become as a contributor to mortality in aging populations. And if we focus specifically on adults age 65 and older, the impact of Alzheimer's disease becomes even clearer. In this age group, Alzheimer's disease rises significantly in the ranking of leading causes of death. While heart disease and cancer still occupy the top positions, Alzheimer's disease becomes one of the most prominent causes of mortality in older adults. This reflects a key biological reality. Alzheimer's is strongly age-dependent. The risk of developing the disease increases dramatically as individuals grow older. As life expectancy increases and more people survive into their 70s, 80s, and 90s, the number of individuals affected by neurodegenerative disease continues to rise. This is why Alzheimer's disease is often described as one of the defining medical challenges of aging populations. Before we focus specifically on Alzheimer's disease, it's important to understand that dementia itself is not a single disorder, but a spectrum of conditions that impair cognitive function. So let's clarify what we mean by cognitive decline and dementia. Cognitive decline is an umbrella term referring to progressive losses in memory, reasoning, attention, and executive function that arise from underlying brain disease. Importantly, dementia is not a single disorder. Instead, it represents a spectrum of neurodegenerative and vascular conditions that impair cognitive function. The most common of these is Alzheimer's disease, which accounts for roughly 60 to 80% of dementia cases worldwide. This disease is characterized by an accumulation of beta amyloid plaques outside neurons and tau protein aggregates inside neurons. Another major contributor is vascular dementia, which results from damaged blood vessels in the brain that reduces oxygen and nutrient delivery to neural tissue. Lewy body dementia involves abnormal aggregation of the protein alpha-synuclein within neurons and often produces both cognitive symptoms and movement abnormalities similar to Parkinson's disease. Finally, frontotemporal dementias primarily affect the frontal and temporal lobes of the brain and often produce early changes in behavior, personality, or language before memory symptoms appear. Together, these disorders represent a heterogeneous group of neurocognitive diseases that progressively impair memory, judgment, and the ability to perform daily activities. Neurodegenerative disease is not limited to disorders that primarily affect memory. Many neurodegenerative diseases affect multiple neural systems and produce both motor and cognitive symptoms. Parkinson's disease provides a good example of this overlap. Parkinson's is the second most common neurodegenerative disorder after Alzheimer's disease and is traditionally associated with motor symptoms such as tremor, muscle rigidity, slowed movement, and a characteristic shuffling gait. These symptoms arise primarily from the degeneration of dopamine-producing neurons in a region of the brain called the substantia nigra, which plays an important role in regulating movement. However, Parkinson's disease also affects cognitive function. As the disease progresses, many patients develop impairments in executive function, attention, and memory. In fact, long-term studies suggest that large proportions of individuals with Parkinson's disease eventually develop dementia over the course of the illness, with estimates approaching 75% or higher. In the United States, approximately 1.1 million people currently live with Parkinson's disease, and about 90,000 new cases are diagnosed each year. So now we'll shift and focus on Alzheimer's disease as a model system for understanding dementia. Alzheimer's disease is widely characterized as a disorder of protein misfolding and disrupted proteostasis, meaning the normal cellular systems that control protein folding, trafficking, and degradation begin to fail. Like several other neurodegenerative diseases, including Parkinson's disease, Huntington's disease, and amyotrophic lateral sclerosis, Alzheimer's involves proteins that adopt abnormal conformations, aggregate, and accumulate within neural tissue. In Alzheimer's disease, two proteins are central to this process: beta amyloid, which accumulates outside neurons to form plaques, and tau, which forms intracellular aggregates known as neurofibrillary tangles. These abnormal protein structures disrupt cellular function, interfere with neuronal signaling, and eventually lead to neuronal damage and loss. Neurons are particularly vulnerable to this type of dysfunction because they are long-lived, metabolically active cells that do not readily regenerate. Unlike many other tissues in the body, damaged neurons cannot simply be replaced through cell division. As a result, small failures in protein quality control can accumulate over time and ultimately contribute to neurodegeneration. The two proteins that are central to the pathology of Alzheimer's disease, amyloid-β, often abbreviated A-beta, and tau. The accumulation of these proteins is considered one of the defining pathological hallmarks of the disease. However, their toxicity does not arise simply from their presence, but from changes in how these proteins are processed, folded, and regulated inside the brain. Amyloid-β accumulates outside neurons, forming structures known as amyloid plaques. Importantly, amyloid deposition can also occur during normal aging. In fact, studies suggest that roughly 20 to 40% of cognitively normal adults show measurable amyloid buildup in the brain. However, in Alzheimer's disease, this accumulation becomes much more extensive and widespread. Nearly 90% of Alzheimer's patients show significant amyloid pathology, often appearing decades before clinical symptoms emerge. Tau pathology remains inside neurons, where tau proteins form twisted aggregates called neurofibrillary tangles. Similar to amyloid, small amounts of tau aggregation can also occur during normal aging. What distinguishes Alzheimer's disease is not simply the presence of tau, but where the tangles appear, how densely they accumulate, and how rapidly they spread through the brain. In Alzheimer's disease, the combination of widespread amyloid deposition and progressive tau pathology ultimately disrupt neural function, leading to synaptic loss, neuronal death, and cognitive decline that characterizes the disease. One way scientists think about protein aggregation in neurodegenerative diseases is by comparing it to prion diseases. Prions are unusual infectious agents composed entirely of misfolded proteins. Unlike viruses or bacteria, they contain no DNA or RNA. Instead, they spread by inducing normal proteins to adopt the same abnormal folded structure. In prion diseases, a normal folded protein, called PRPC, can convert into a misfolded form known as PRPSC. Once this misfolded protein appears, it can interact with normal proteins and cause them to misfold. This creates a chain reaction where misfolded proteins accumulate and progressively damage brain tissue. These diseases are called transmissible spongiform encephalopathies. Examples include Creutzfeldt-Jakob disease in humans and bovine spongiform encephalopathy or mad cow disease in cattle. While Alzheimer's disease is not infectious like prion diseases, researchers have discovered that both amyloid-β and tau proteins can behave in a similar way, where misfolded proteins act as templates that promote additional protein misfolding. This prion-like propagation may help explain how pathological proteins spread through brain networks during the progression of Alzheimer's disease. The protein that gives rise to amyloid beta is called the amyloid beta precursor protein, or APP. APP is a single-pass transmembrane protein that is widely expressed in neurons and many other cell types. Importantly, APP itself is not inherently toxic. The pathological risk arises from how the protein is processed by cellular proteases. Because APP spans the membrane, its processing is influenced by factors such as membrane composition, the lipid environment, and intracellular trafficking. This illustrates an important biochemical principle. The function and fate of a protein depends not only on its sequence, but also on its cellular context. APP can be processed through two competing enzymatic pathways. In the non-amyloidogenic pathway, shown on the left, APP is first cleaved by enzymes from the Adam family of metalloproteases, which function as alpha secretases. This cleavage occurs with the amyloid beta region of APP, which prevents amyloid formation. The reaction releases a soluble extracellular fragment called SAPP alpha and leaves behind the membrane-associated fragment called C83. In the amyloidogenic pathway, shown on the right, APP is first cleaved by the beta secretase, also known as base 1. This cleavage generates a longer membrane fragment called the C99 fragment and releases SAPP beta outside the cell. The C99 fragment is then cleaved by gamma secretase, a multi-protein complex composed of presenilin, nicastrin, PEN2, and APH1. This second cleavage releases amyloid beta, which can accumulate outside neurons and eventually form amyloid plaques. At the same time, the APP intracellular domain, or AICD, is released into the cytoplasm of the cell. Amyloid beta, or A-beta, refers to a family of peptides produced from APP that range from approximately 36 to 43 amino acids in length. Structurally, A-beta is best described as an intrinsically flexible or conditionally folded peptide, meaning that its structure depends strongly on its environment. Factors such as membrane surfaces, peptide concentration, interaction with lipids or metal ions, or other A-beta molecules can influence the conformations that the peptide adopts. Under normal physiological conditions, A-beta exists as a dynamic ensemble of structures, often containing alpha helices or random coil conformations. In this state, the peptide is relatively soluble and less prone to aggregation, and it can be efficiently cleared by cellular proteases and immune cells such as the microglia. In this sense, the alpha helical and disordered conformations can be thought of as kinetically protective because they reduce the likelihood that A-beta molecules will aggregate. However, under certain conditions, the peptide undergoes a conformational transition to a beta sheet-rich structure. This shift promotes interactions between neighboring peptides and allows them to stack together into oligomers and eventually amyloid fibrils. Several factors can promote this structural transition, including increased A-beta concentration, impaired clearance, interactions with membrane surfaces or metal ions, and enrichment of longer A-beta 42 peptides, which is particularly prone to aggregation. Amyloid beta peptides vary in length, just as we said on the last slide, depending on where the gamma secretase complex cleaves the APP protein. The two most common forms are A-beta 40 and A-beta 42. Although A-beta 40 is actually produced in larger amounts, the form that is most strongly associated with disease is A-beta 42. In amyloid plaques, the ratio of these peptides shift dramatically. A-beta 42 becomes disproportionately enriched and forms the structural core of the plaque, while A-beta 40 is more commonly found towards the peripheral regions of the fibril structure. The reason for this difference comes down to two additional amino acids at the C terminus of the A-beta 42. These residues are hydrophobic, which significantly changes the peptide's aggregation behavior. These extra hydrophobic residues have three important effects. They increase the peptide's tendency to adopt beta sheet structures. They stabilize early oligomer nuclei, which act as seeds for aggregation, and they lower the energetic barrier for fibril formation. As a result, A-beta 42 aggregates much more readily than A-beta 40. It typically forms the initial nucleation core, after which other A-beta peptides, including A-beta 40, are recruited into the amyloid fibril. In this way, A-beta 42 acts as the aggregation trigger, while other A-beta species contribute to plaque growth. Amyloid beta is produced when the beta secretase and gamma secretase enzymes cleave the APP protein in the amyloidogenic pathway. Because the gamma secretase cleavage site is somewhat imprecise, several A-beta peptide lengths can be produced, with the two most common, the A-beta 40 and A-beta 42. Once released, these A-beta peptides exist initially as disordered monomers in solution. However, when their concentration increases or clearance becomes impaired, they begin to self-associate through a nucleation-dependent aggregation process. The kinetics of this process typically follow a sigmoidal growth curve with three phases. The first phase is a lag phase where soluble A-beta monomers accumulate and slowly form a small nucleation seed. Once this nucleus forms, aggregation accelerates dramatically during the elongation phase, where additional monomers rapidly assemble into oligomers, protofibrils, and eventually fibrils. Finally, the system reaches a saturation phase when fibril growth slows as the available monomer pool becomes depleted. As shown in the diagram, A-beta aggregation can proceed through multiple structural pathways. In the on-pathway route, monomeric A-beta forms oligomers, which then assemble into protofibrils and mature fibrils that ultimately develop into the amyloid plaques. Alternatively, aggregation can occur through off-pathway routes where monomers assemble into amorphous aggregates that do not efficiently form fibrils. Recently, work also suggests that A-beta oligomers can undergo liquid-liquid phase separation, forming liquid droplets or biomolecular condensates. These condensates can act as concentrated environments that promote fibril formation as they mature. Over time, fragmentation of fibrils can generate new aggregation seeds, allowing plaques to expand and propagate through the surrounding tissue. One important feature of amyloid biology is that amyloid fibrils are not structurally uniform. Instead, amyloid beta plaques can exist in multiple polymorphic forms. This polymorphism arises because the A-beta peptide can fold and pack into different beta sheet architectures during aggregation. Even small differences in peptide sequence or aggregation conditions can lead to different fibril structures, including differences in beta strand organization, monomer packaging, fibril symmetry, and overall fibril morphology. The structures shown here illustrate two different fibril architectures determined for the A-beta 40 fibrils. Although both are composed of stacked beta sheets, the peptides pack together differently, producing distinct fibril morphologies. And importantly, A-beta 42 tends to show even greater conformational heterogeneity than A-beta 40 because its additional hydrophobic residues promote multiple aggregation pathways. This structural diversity creates major challenges for molecular imaging, structural determination, and therapeutic targeting because many different amyloid structures may interact differently with antibodies, imaging probes, or small molecule inhibitors. At present, researchers are still working to determine how these different amyloid polymorphs relate to disease progression and toxicity. So as we've stated, amyloid plaques are one of the defining pathological features of Alzheimer's disease. However, an important discovery in the field is that the total amount of plaque in the brain correlates only weakly with cognitive decline. Instead, current research suggests that the most neurotoxic species are soluble beta oligomers rather than the larger insoluble plaques themselves. As shown in the diagram, amyloid beta peptides exist along a spectrum of aggregation states. Monomeric peptides can assemble into small soluble oligomers, which can then grow into larger oligomers, protofibrils, fibrils, and eventually the amyloid plaques. The earlier assemblies, particularly the small soluble oligomers, are now believed to be the most disruptive to neuronal function. These oligomers are highly dynamic and structurally flexible, which allows them to interact with neuronal membranes and synaptic receptors. Through these interactions, oligomers can disrupt calcium homeostasis, alter membrane permeability, and interfere with synaptic signaling, ultimately contributing to synaptic loss and neuronal dysfunction. This distinction highlights an important principle in protein misfolding diseases. Intermediate or transient protein assemblies are often more biologically toxic than the large stable aggregates that eventually accumulate. In addition to amyloid pathology, tau protein plays a central role in Alzheimer's disease. Tau is a microtubule-associated protein, meaning that its normal function is to bind and to regulate microtubules within neurons. Microtubules are long protein filaments that form part of the cytoskeleton, providing both structural support and intracellular transport pathways within the cell. In neurons, these structures are especially important because materials such as neurotransmitters, vesicles, and proteins must be transported over very long distances along the axon. Tau helps regulate this system by binding to microtubules and stabilizing their structure. Structurally, tau is an intrinsically flexible protein that behaves largely like a random coil in solution, meaning it doesn't adopt a single rigid folded structure. This flexibility allows tau to interact dynamically with microtubules and other proteins. When tau binds to the surface of the microtubules, it can influence microtubule stability and spacing, which in turn affects how motor proteins interact with them. For example, motor proteins such as kinesin move along microtubules to transport cargo down the axon terminal. Tau can regulate this transport by influencing how accessible the microtubule surface is to these motor proteins. In this way, tau plays an essential role in maintaining neuronal structure and regulating intracellular transport. One important feature of tau protein is that it contains a large number of potential phosphorylation sites. This is because the tau sequence contains many serine, threonine and tyrosine residues, which can be modified by cellular kinases. Remember that kinases are enzymes that phosphorylate their downstream target. In the largest tau isoform, there are dozens of potential phosphorylation sites, including more than 45 serine residues and 35 threonine residues that can be targeted. Tau is also intrinsically flexible, which means that in solution, it adopts an open and dynamic structure. This flexibility makes many of the residues accessible to kinases, allowing tau phosphorylation to be tightly regulated under normal conditions. In healthy neurons, tau is moderately phosphorylated, primarily on specific serine and threonine residues. These normal phosphorylation events help regulate how tau interacts with microtubules. However, in Alzheimer's disease, tau becomes abnormally hyperphosphorylated. As shown in the diagram, additional phosphorylation sites become modified in the disease state. The green boxes here represent phosphorylation sites that are commonly observed under normal physiologic conditions, while the red boxes indicate additional phosphorylation sites that become modified in Alzheimer's disease. These modifications change the charge distribution along the tau protein, weakening its ability to bind to microtubules. As a result, tau detaches from the microtubules, leading to both a loss of its normal stabilizing function and an increased tendency for tau molecules to self-associate and aggregate. This process reflects an important theme in neurodegenerative diseases, a combination of loss of normal protein function and toxic gain of function through aggregation. The hyperphosphorylation of tau is thought to arise from dysregulation of kinase and phosphatase activities, highlighting the important role that post-translational modifications play in regulating protein behavior. Once tau detaches from microtubules, the protein becomes more prone to forming paired helical filaments, which ultimately assemble into neurofibrillary tangles. So when tau becomes abnormally hyperphosphorylated, its ability to bind to microtubules is weakened, and as tau detaches from the microtubules, the protein becomes much more prone to self-aggregation and association. One of the characteristic structures formed by aggregated tau is known as the paired helical filament, or PHF. The electron micrographs shown here illustrate these structures. On the left, we see paired helical filaments that were isolated directly from the brains of Alzheimer's disease patients. On the right, we see similar filaments generated in vitro using recombinant tau protein. The in vitro tau protein contains a mutation that promotes aggregation. These filaments have a distinct twisted ribbon-like structure with repeating crossover points roughly every 80 nanometers, which can be seen in the arrowheads at this image. As the filaments accumulate within the neurons, they assemble into larger structures known as neurofibrillary tangles, which are another major pathological hallmark of Alzheimer's disease. Like amyloid-β fibrils, tau filaments are thought to adopt beta sheet-rich structures, although determining their precise molecular architecture has been a major challenge in the field. So this diagram summarizes the progression of tau pathology in Alzheimer's disease. In healthy neurons, tau protein binds to microtubules and helps stabilize the cytoskeleton, which is essential for intracellular transport along the axon. In Alzheimer's disease, tau becomes abnormally hyperphosphorylated, which weakens its interaction from the microtubules. As tau detaches, the microtubules become destabilized. Once free in the cytoplasm, tau proteins begin to self-assemble, first forming dimers, and then small oligomers, which are believed to be some of the most toxic species. These oligomers then assemble into paired helical filaments, or PHFs, which have characteristic twisted structure. Over time, many of these filaments will accumulate together into bundles and larger aggregates called neurofibrillary tangles. Unlike the amyloid-beta plaques, which form outside neurons, tau tangles develop inside neurons where they disrupt cellular transport systems and contribute directly to neuronal dysfunction and cell death. Importantly, the distribution and amount of tau pathology correlates strongly with cognitive decline, making tau aggregation a key driver of disease progression in Alzheimer's disease. This slide illustrates how tau pathology progresses over time. Normally, tau exists as soluble monomers inside neurons, where it stabilizes microtubules. However, when tau becomes misfolded or aggregated, it can begin to form small fibrillary structures. These fibrils can then act as seeds, recruiting normal tau proteins and converting them into the same misfolded structure. This process drives the formation of larger aggregates, including paired helical filaments and eventually the neurofibrillary tangles. One important hypothesis for how tau pathology spreads through the brain is called prion-like propagation. In this model, small tau aggregates, or preformed fibrils, often abbreviated as PFFs, can move from one neuron to another. When these seeds enter a neighboring cell, they can template the misfolding of normal tau proteins, causing additional aggregation. Experimental studies have shown that introducing very small amounts of tau fibrils into cells or animal models can trigger widespread tau aggregation, supporting this seeded propagation mechanism. As a result, tau pathology spreads through the brain in a predictable pattern along connected neural circuits, which closely matches the progression of symptoms observed in Alzheimer's disease. Importantly, the term prion-like refers only to this template protein misfolding mechanism. Unlike true prions, tau pathology is not considered infectious between individuals. So to summarize this lecture, Alzheimer's disease can be understood as a disorder of protein chemistry and protein homeostasis. The disease appears to begin with the misprocessing of the amyloid precursor protein, which leads to the accumulation of amyloid-beta peptides. These peptides can assemble into soluble oligomers and eventually form amyloid plaques. While amyloid accumulation appears to initiate the pathological cascade, the progression of the disease correlates much more strongly with tau pathology. When tau becomes abnormally modified and misfolded, it detaches from microtubules and aggregates into paired helical filaments and neurofibrillary tangles, disrupting neuronal function and contributing to cell death. Together, these processes illustrate how disruptions in protein folding, processing, and post-translational regulation can overwhelm the ability of neurons to maintain protein homeostasis. These molecular events ultimately trigger broader cellular failures, including synaptic dysfunction, inflammation, and neuronal loss, which we will explore in the next lecture.